Hi everyone, this is Yinan from ACROBiosystems. Thank you very much for having me here. It's a great honor for me to have this opportunity to share with you the critical role of antigens and antibodies in SARS-CoV-2 serological test development. In this webinar, I would like to focus on what ACROBiosystems has done to help fight against COVID-19. Now, I will start my presentation with the introduction of the outbreak of COVID-19. COVID-19 pandemic is caused by the emerging coronavirus SARS-CoV-2, and it presents a serious global public health emergency. The first cases of COVID-19 were reported from Wuhan, China, in December 2019. As of August 26th, over 24 million confirmed cases of COVID-19 have been reported, and over 0.8 million deaths have been caused by this pandemic. Here shows the COVID-19 map offered by Johns Hopkins Coronavirus Resource Center. The red dots represent the confirmed cases of COVID-19, and the larger the red dots, it means the more confirmed cases in the specific region or country. And you can see that COVID-19 has spread to most parts of the globe. In addition to the health damage caused by this pandemic, COVID-19 has huge impact on our daily life. Now we have got used to wearing protective masks and keeping social distance in the public place. COVID-19 is caused by SARS-CoV-2, which belongs to coronavirus. Now let's take a look at the background of this virus species. Coronavirus was first identified by Dr. Almeida in 1964 in St. Thomas Hospital in London. They are a group of enveloped viruses that have a halo or crown-like appearance when viewed under an electron microscope. The genome of coronaviruses are composed of positive-sense single-stranded RNA, and their genome size ranges between 27 to 34 kilobases, which is relatively large for RNA viruses. Coronavirus infect mammals and birds and cause various symptoms such as respiratory tract disease and diarrhea. This virus species can be categorized into 4 genome groups, including alpha, beta, gamma, and delta. And within the beta coronavirus genome group, there are 4 lineages, including A, B, C, and D. 7 coronaviruses have been reported to be able to infect humans. Among them, HCOV229E, HCOV-NL63 belong to the alpha genome groups, and HCOV-HKU1, HCOV-OC43 belong to A lineage of beta genome group. These 4 coronaviruses can only cause common cold. SARS-CoV and SARS-CoV-2 belong to B lineage, and MERS-CoV belong to C lineage of beta genome group. These 3 coronaviruses have been shown to be lethal. At present, the COVID-19 pandemic within many countries have not yet reached its peak, and the number of cases and deaths are predicted to rise in the coming weeks and months. Early and accurate diagnosis of COVID-19 is essential, not only to ensure appropriate patient care, but also to facilitate identification of SARS-CoV-2 infected people to control disease spread. Now, there are 3 types for SARS-CoV-2 diagnosis, which are molecular tests, antigen tests, and antibody tests. The application of the 3 methods are a bit different, and they have their own advantage and disadvantage. For molecular test, it is typically highly accurate and can be used to diagnose active coronavirus infection, but it requires equipment and laboratories to conduct the experiment, and it usually takes 1 to 3 days to get the results. For antigen test, it can be used to diagnose acute infection, and it only takes 15 to 30 minutes to get the results, but sometimes it may give out a false negative outcome. For antibody test, it shows if you have been infected by this virus in the past, and it also gives results very quickly as antigen test. However, antibody tests cannot diagnose active infection at the time of test or show that you do not have COVID-19. Since SARS-CoV-2 vaccine is expected to be launched at the end of this year, then people who have been vaccinated will have anti-SARS-CoV-2 antibodies in their body, and the antibody test will not be able to diagnose whether they have been infected by SARS-CoV-2. The molecular test and antigen test will be the main diagnosis method to test the acute infection of SARS-CoV-2. Up until now, 17 SARS-CoV-2 antibody detection kits have been approved by FDA. However, only 2 SARS-CoV-2 antigen detection kits, which are produced by BD and Quidel, have received the emergency use authorization from FDA. In my opinion, more antigen tests will be approved by FDA in the next few months. After talking about the comparison between different SARS-CoV-2 infection methods, I will be introducing what ACROBiosystems has done to help fight against COVID-19. First, I would like to offer you a brief introduction of ACROBiosystems. ACROBiosystems is founded in 2010, and we are a leading manufacturer of recombinant proteins and other critical reagents to support the development of targeted therapeutics. ACROBiosystems is dedicated to accelerating the process of target therapeutic drug development from the starting point of target identification to endpoint of clinical trial. ACROBiosystems has served over 4,500 customers from over 60 countries and reached out to 20,000 end users from pharmaceutical companies and academic institutes. ACRO has established long-term collaboration with big pharma such as Pfizer, Novartis, Johnson & Johnson, and well-known academic institutes like NIH. To provide better service to end users, ACROBiosystems has set up 2 working places at Beijing and Shanghai in China, and 3 offices at Boston, San Jose, and Newark in US. With the outbreak of COVID-19 in China, ACROBiosystems responded quickly and finished the design of SARS-CoV-2 antigen products during Chinese Spring Festival, and immediately started to produce the antigen after getting back to work. Up until now, we have launched over 80 SARS-CoV-2 antigen and antibody products, and another 20 products are under development. The majority of these antigen products are spike ectodomain, S1 subunit, and RBD. Also, the antibody products including anti-nucleocapsid protein and anti-S1 antibodies. ACRO has worked hard on developing products that can be used in antigen test and antibody test development. We have made following efforts to provide high-quality products to our customers and help fight against COVID-19, including verifying the trimeric state of our spike ECD protein product, the production of biotinylated spike protein, optimizing the expression construct for spike protein RBD, producing mutant spike S1 and RBD protein, and wild-type nucleocapsid protein from HEC293 cells, and lastly, developing anti-SARS-CoV-2 antibodies. First, let's see how ACROBiosystems produce SARS-CoV-2 spike protein in trimeric state. SARS-CoV-2 spike protein is a key transmembrane glycoprotein that forms homotrimers protruding from the viral surface and mediate host cell attachment and cell entry. It was reported that SARS-CoV-2S and SARS-CoV-S share the same functional host cell receptor, which is a angiotensin converting enzyme 2 or ACE2. Spike protein contains 1,273 amino acids and can be cleaved into S1 and S2 subunits. S1 subunit is responsible for binding to the host cell receptor. It consists of N-terminal domain or NTD and receptor binding domain or RBD. S2 subunit mediates fusion of the viral and cellular membranes, which contains fusion peptide, peptide repeat 1, and central helix. Currently, spike protein extracellular domain, including both S1 and S2 subunits, S1 subunit, and RBD, have been widely used in the serological test development. We'll talk about the application of these antigens into details later. The trimeric state of spike protein is required to develop a serological test kit for COVID-19. NIH has published a paper describing the development of an ELISA kit for several surveys of the SARS-CoV-2 pandemic. In that paper, the trimeric structure of SARS-CoV-2 spike protein was first verified by analytical size exclusion chromatography and transmission electron microscopy, and then deployed in the ELISA assay. To produce trimeric spike protein, we introduced a GS-AS substitution at the S1-S2 fusion cleavage site to maintain the integrity of spike protein. In addition, a trimerization domain was introduced just in the upstream of the predicted transmembrane region to facilitate the trimer formation of spike protein. We have launched 2 spike trimer protein products, including spike trimer protein from the original version of SARS-CoV-2 and spike trimer protein with D614G mutation in the sequence, which becomes popular now in Europe and North America. ACROBiosystems uses its proprietary HAC293 cell expression platform to produce this recombinant spike protein trimer. The purity of this protein is greater than 90% in SDS page, and the protein migrates as about 200 kilodaltons under reducing conditions due to glycosylation. Since the purity on SDS page cannot indicate the conformational homogeneity of the protein, we perform SAC-MOS to show that the trimeric S protein is with more than 80% purity and with molecular weight around 520 to 620 kilodaltons. To further characterize the bioactivity of the spike protein, we use surface plasmon resonance to measure the binding ability between spike trimer and its host receptor ACE2. The constant dissociation shows around 35 nanomolar, which is consistent with the binding affinity reporting paper. In addition, we are the first company around the world that provide the negative stain electron micrograph for our spike trimeric protein. The results shows that the well-folded trimeric particles of SARS-CoV-2 S protein are similar in size and appearance to previously characterized SARS-CoV-2 trimers. We also performed SAC-MOS to compare our SARS-CoV-2 spike protein product with the protein offered by other vendors. As you can see from this slide, in contrast to ACRO's trimeric spike protein product with conformational purity of 97%, the spike protein offered by other vendors are in high polymer form with molecular weight around 4,500 kilodaltons. Now, I will introduce the production of biotinylated spike protein by ACROBiosystems. In addition to unconjugated antigen product used in serological test kits, the biotinylated SARS-CoV-2 recombinant antigen is also employed in commercial chemiluminescent immunoassay products. For example, on May the 29th, Siemens Healthineers Diagnostics receives FDA emergency use authorization for its SARS-CoV-2 total antibody test. The total antibody test has demonstrated 100% sensitivity and 99.8% specificity in identifying SARS-CoV-2 antibodies. The antigen used in the antibody test is biotinylated SARS-CoV-2 recombinant antigen, which bind the streptavidin-coated microparticles and capture anti-SARS-CoV-2 antibodies in the specimen. ACROBiosystems has developed an exclusive collection of pre-labeled and experimentally validated biotinylated SARS-CoV-2 spike proteins. These products are produced using the AVI-Tec technology, which introduces a unique 15 amino acid peptide AVI-Tec into the recombinant protein during expression vector construction. The single lysine residue in the AVI-Tec is enzymatically biotinylated by the E. coli biotin ligase A. In this way, the biotinylation only happens on the single point lysine residue of AVI-Tec. The protein orientation is uniform when immobilized on a streptavidin-coated surface. One of our customer drugs and diagnostics for tropical diseases has commented after using our biotinylated spike proteins that ABATech biotinylation technology from ACROBiosystems is a great fit for our LFA platform. We have launched 3 biotinylated SARS-CoV-2 related products, including spike trimer protein, S1 protein, and RBD protein. Biotinylated spike trimer protein with D614G mutation and biotinylated nuclear capsid protein will be launched soon. ACRO's biotinylated SARS-CoV-2 antigen products are also used to detect vaccine-induced antibody titers. In a paper published by Pfizer and BioNTech, they reported the results of the phase 1 study of their mRNA vaccine with the name of BNT162b1. In this study, the researchers used ACROBiosystems biotinylated SARS-CoV-2 RBD protein to detect the RBD binding IgG concentrations from 10 or 30 microgram dose recipients. It's shown that RBD binding IgG concentrations were detected at 21 days after the first dose and substantially increased 7 days after the second dose given at day 21. After dose 2, with a 10 microgram or 30 microgram BNT162b1, the RBD binding IgG GMCs were around 8-fold or around 50-fold that of the convalescent serum panel GMC, which means the vaccine candidate can induce robust immune response to SARS-CoV-2. Next, we'll discuss the importance of expression construct for RBD in serological test development. Recently, a paper published by NIH has shown that different RBD constructs have distinct signal when used in developing serological ELISA assay. The 2 RBD constructs from Kramer lab of Mount Sinai and the Reagan Institute were compared, and the construct from Reagan Institute displayed stronger signal with a recombinant RBD antibody. ACRO also worked hard on the optimization of RBD expression constructs. We have tested tens of different constructs and have found out several constructs that can be used in serological test development, which can increase the sensitivity while maintain the specificity of the test kits. These products have been launched on our website. In the fourth part, we'll talk about the production of mutant spike S1 and RBD proteins. ACRO noticed that several RBD mutants have been reported in some strains of SARS-CoV-2, and we generated the mutant protein products to characterize the difference between wild-type and mutant RBDs. ELISA and its SPR results have shown that RBD mutants displayed in this table can bind ACE2 with 2 to 6-fold higher affinity compared with wild-type RBD. These mutant RBDs can be used to test the neutralizing ability of anti-SARS-CoV-2 antibodies to exclude the possibility that the mutation in RBD makes the antibody unrecognizable, makes the protein unrecognizable for the antibody. We tested the binding ability between our anti-SARS-CoV-2 antibodies and the RBD mutants. These antibodies all show similar binding affinity with RBD mutants and the wild-type RBD in SPR assay. In addition, the antibodies can effectively inhibit the interaction between RBD and ACE2. These studies indicate that RBD mutants developed by ACROBiosystems are useful research reagents to verify the broad-spectrum neutralizing antibodies against different strains of SARS-CoV-2. In addition, we also developed S1 protein with D614G mutation. Next, I would like to introduce how ACROBiosystems has improved the performance of nuclear capsid protein in LFA test development. Nuclear capsid protein participates in RNA package and virus particle release. Therefore, it is an important antigen for coronavirus and it is frequently used in serological assays. SARS-CoV-2 N protein contains two distinct RNA binding domains, the N-terminal domain or NTD and the C-terminal domain or CTD, linked by a poorly structured linkage region LKR. However, the high isoelectric point and oligomerization of N protein makes it difficult to be modified by colloidal gold in LFA test development. ACROBiosystems has tried multiple methods to increase the performance of N protein in LFA test development. For example, we optimize the expression constructs and screen the expression system for N protein. Finally, we found that full-length N protein produced in HEC293 cells performs well in colloidal gold modification and increases the specificity compared to N protein expressed from prokaryotic cells. Now, this product is a very popular protein among our product pipeline. In the last part, I will talk about how ACROBiosystems has developed anti-SARS-CoV-2 antibodies that can be used in antigen and antibody test development. In addition to offer SARS-CoV-2 antigen products, ACROBiosystems has collaborated with its partners to develop anti-SARS-CoV-2 antibodies as well. We use our high-quality SARS-CoV-2 antigen products to immunize mouse and obtain multiple anti-SARS-CoV-2 antibodies. Taking advantage of the ELISA assay, we conducted rapid screening of antibodies and selected the candidates that have high binding affinity towards SARS-CoV-2 antigens. Next, we utilized SPR assay to measure the KD value for each antibody and map the epitope of these antibodies. As you can see here, we have categorized the antibody candidates into 6 groups, and in each group, the antibody will compete with the others in antigen binding. After screening and epitope mapping, we have selected 5 anti-N protein antibodies and 4 anti-S1 antibodies for our customers to develop antigen tests. Here shows the list of our anti-SARS-CoV-2 antibody products. We also tested the sensitivity of each antibody pairs in ELISA and LFA assays. This table shows the matched antibody pairs and the recommendation for capture or detection use in ELISA. Since we are not sure if these antibody pairs can fit our customer's R&D platform, it should be noted that the matched antibody pairs listed here is only for reference. For NUNS46 and NUNS47 antibody pair, the sensitivity of detecting nucleocapsid protein is 50 picogram in our ELISA assay and 12 picogram per milliliter in ELISA assay, respectively. We tested the species specificity of this antibody pair and found that NUNS46 and NUNS47 antibody pair can only recognize SARS-CoV-2 nuclear capsid protein, but not nuclear capsid protein from other coronaviruses. Similarly, we also measured the sensitivity of detecting S1 protein for S1AM12A1 and S1NM13A1 antibody pair. The sensitivity is 200 picogram in our ELISA assay and 24 picogram per milliliter in ELISA assay, respectively. This antibody pair can only recognize SARS-CoV-2 S1 protein, but not S1 protein from other coronaviruses. For serological test development, ACROBiosystems has launched both anti-SARS-CoV-2 S1 IgG and IgM antibodies. SAD-S35 is isolated from SARS-CoV-2 infected patients, and it combined to S1 protein with sensitivity of 0.1 nanogram per milliliter. In LFA assay, the sensitivity of this antibody is 0.5 nanogram. SPD-M141 is a chimeric antibody combining the constant domains of the human IgG molecule with mouse variable regions. It can bind to S1 protein with sensitivity of 0.2 nanogram per milliliter. In LFA assay, the sensitivity of this antibody is 1 nanogram. To conclude this talk, ACROBiosystems has developed SARS-CoV-2 antigen and antibody products specifically designed and optimized for serological test development. Spike-derived proteins are produced from HEC293 cells, while nucleocapsid proteins are either yielded from HEC293 cells or E. coli cells. The antigens are expressed with a variety of tags, including his, human FC, mouse FC, and his AVI tag, which could meet the needs of colloidal gold immunoassay, chemiluminescence immunoassay, and ELISA development. If you are interested in any of these products, please contact us for more detailed information. Now, this will be the last slide of our presentation. Thanks everyone for attending today's webinar. We're doing a raffle for everyone. We will have the chance to win our adorable llama, the very popular among our customers. If you have not already entered this raffle, you can go to our website www.acrobiosystems.com and enter it. We will contact you within 3 business days if you win the llama. Okay, that's all for my presentation. And now I would like to answer the questions from the audience.